Good afternoon to one and all. And my topic for seminar is gene therapy and barriers of gene therapy. What is gene therapy? Gene therapy is the use of genes as a medicine. This process includes gene inactivation, gene repair, and gene surgery. This technique involves replacement of faulty genes with the help of healthy genes to fight against disease. Gene therapy can be accomplished by replacing mutated gene that causes disease with a healthy copy of the gene, inactivating or knocking out a mutated gene that is functioning improperly, introducing a new gene into the body to help fight a disease. The first approved gene therapy experiment occurred on September 14, 1990 in US when Ashanti De Silva was treated for adenosine deaminase SID, severe combined immunodeficiency. Process of gene therapy: identification of the defective gene, cloning of normal healthy gene, manipulation of vector, and insertion of therapeutic gene into the vector. Vector is inserted into target tissue or organ of the patient. Vector transfers genetic material with therapeutic gene into the cell. Functional protein is produced which restores target cells to a normal state. Types of gene therapy: somatic cell gene therapy and germline gene therapy. In somatic cell gene therapy, the therapeutic genes are introduced into the somatic cells. Example, introduction of genes into the bone marrow cells, skin cells, blood cells, etc. It will not be inherited in later generations. Germline gene therapy. In germline gene therapy, the therapeutic genes are introduced into the germ cells. Example, introduction of genes into eggs and sperms. It is heritable and passed on to later generations. Due to safety and ethical reasons, it is not being attempted at present. Somatic cell gene therapy is further divided into ex vivo gene therapy and in vivo gene therapy. Ex vivo gene therapy. The ex vivo gene therapy can be applied to only selected tissues. Example, bone marrow, whose cells can be cultured in the laboratory. Steps in ex vivo gene therapy: isolate cells with genetic defect from a patient, grow the cells in culture, introduce therapeutic gene to correct defect gene. Select the genetically corrected cells and grow. Transplant the modified cells to the patient. Example of ex vivo gene therapy. First, gene therapy to correct deficiency of adenosine deaminase. Deficiency of enzyme adenosine deaminase was performed on a 4-year-old girl, Ashanti De Silva, who was suffering from severe combined immunodeficiency caused due to gene coding for ADA. Due to the presence of ADA in the bone marrow will synthesize B and T cells and the immune system will start working. But in a 4-year-old girl, there is no ADA in the bone marrow. So, the bone marrow will not synthesize B and T cells and there is no immune response, and this condition is known as SID. To treat this, the ADA gene is introduced into the lymphocytic system with the help of retrovirus. Further, in the bone marrow, the B and T cell will start producing. Next, in vivo gene therapy. In vivo gene therapy, the therapeutic gene is directly delivered into the target tissue of the patient's body. It is carried out by viral or non-viral vector systems. Example of in vivo gene therapy. Therapy for cystic fibrosis. In patients with cystic fibrosis, a protein called cystic fibrosis transmembrane regulator is absent due to the gene defect. Due to the absence of this gene, this leads to the accumulation of thick mucus in the respiratory tract and lungs. Here, the normal human gene is cloned in the bacterium and the normal gene is inserted into the harmless virus, adeno-associated virus, and the virus is dripped into the lung through a thin tube inserted into the nose. Virus enters the lung cell nucleus. This is the lung cell and this is the nucleus. The normal gene is inserted into the lung cell DNA by virus. Cells now with normal genes function normally. Barriers to gene therapy. Gene delivery and activation, immune response, disrupting important genes in target cells, commercial viability. Gene delivery and activation. During gene therapy, gene must reach to its destination and it must be activated. Delivering a gene to the raw tissue would be inefficient and it could cause health problems for the patient. Immune response. Our immune system is very good at finding foreign molecule. Immune response is the very important challenge during gene therapy. This situation can be overcome by using immunosuppressive drugs to temporarily suppress the immune system. Disrupting important genes in target cells. Ideal gene therapy is that an introduced gene will continue working for the rest of the patient's life. When we introduce foreign gene, it alters the target cell genome and introduce new challenges. Commercial viability. Some diseases that can be treated with gene therapy, such as cancer, are much more common. Many genetic disorders that can be potentially be treated with gene therapy are extremely rare, some affecting just 1% out of a million. Some gene therapy, including taking in clinical trials necessary for government approval, which are very expensive. Vectors in gene therapy. the carrier particles or molecules used to deliver genes to somatic cells are referred to as vectors. Many gene therapy clinical trials rely on retroviruses or adenoviruses to deliver the desired gene. Other viruses used as vectors include adeno-associated viruses, lentiviruses, pox viruses, alpha viruses, and herpes virus. Retrovirus vector system. Retrovirus are a class of enveloped viruses that contain a single-stranded RNA genome. They are able to efficiently integrate permanently into the human genome. Retroviral vectors are created by removal of the retroviral GAL, POL, ENA genes. These are replaced by the therapeutic genes. Adenoviral vector system. Adenoviruses are medium-sized, non-enveloped icosahedral viruses composed of nucleocapsid and double-stranded linear DNA genome. Adenoviruses are able to deliver large DNA particles, but in contrast to retrovirus, as they would not integrate into the host genome, their gene expression is too short-term. Modified DNA injected into the vector. Vector binds to cell membrane. Vector is packaged in vesicle. Vesicle breaks down releasing vector. Vector injects new gene into the nucleus. Cell makes protein using new gene. Adeno-associated virus. Adeno-associated virus from the parvovirus family are small viruses with a genome of single-stranded DNA. This type of virus is being used because it is non-pathogenic. These viruses can insert the genetic material at a specific site on chromosome 19. Lentivirus vector system. Lentivirus vectors are a subclass of retroviruses. These viruses naturally integrate with non-dividing cells. Non-viral delivery. Pure DNA constructs. This is the simplest method in which pure DNA constructs is directly introduced in the target tissue. The efficiency of DNA uptake by the cells and its expression is rather low. Lipoflexes. The lipid DNA complexes are referred as lipoflexes. They add a DNA construct surrounded by artificial lipid layers. They are non-toxic and non-immunogenic. Other types of gene therapy includes gene augmentation therapy and gene inhibition therapy. In gene augmentation therapy, it is used to treat where a mutation in the DNA causes a loss of the function of a gene. Gene augmentation therapy adds DNA contained in a functional version of this gene back into the cell. Example, replacement of defective P53 gene by normal one in liver cancer. Gene inhibition therapy. Gene inhibition therapy aims to reduce or stop the activity of the problematic gene. This is particularly useful in cancers which are often caused by the overexpression of genes that promote the cell growth and division. It includes two types, antigen and antisense. Antigen blocks transcription with the help of antigen oligonucleotides. Antisense blocks translation with the help of antisense oligonucleotides. Advantages of gene therapy. Gene therapy has the potential to eliminate and prevent hereditary diseases such as cystic fibrosis, AIDS, ADA, SID, etc. It is possible to cure heart diseases, AIDS and cancer. It can be used to eradicate disease from the future generation. Disadvantages of gene therapy. Viruses used as vectors for gene transfer may cause toxicity, immune responses and inflammatory reactions in most disorders caused by defects in multiple genes cannot be treated effectively